So this video is for those of you who need to review the macromolecules, their structure, function, subunits, and things like that. So for each of these questions, feel free to pause the video and try it yourself. Once you have the answer, play the video to see if it's correct. So let's start with this one. Number 1. Name the 4 types of macromolecules. So the first one are carbohydrates. These macromolecules have the empirical formula CH2O. The second type are lipids. These include your triglycerides, your fatty acids, your steroids, phospholipids, and things like that. The third type are the proteins. And the fourth type of macromolecule that you need to be aware of are nucleic acids. So those are the 4 main biological molecules that you need to know for the test you're studying for. Number 2, which of the following carbohydrates is a monosaccharide? A monosaccharide is basically a monomer of a carbohydrate. It's the smallest subunit of a carbohydrate. And glucose is a monosaccharide. Here is the structure of glucose. Glucose is a 6-carbon sugar, and it forms a 6-membered ring. So this is carbon 1, 2, 3, 4, 5, and 6. And the other atoms are hydrogen atoms. Glucose is an aldohexose. Whenever you hear the end in -ose, it tells you it's a carbohydrate. Now when you think of hex, hex means 6. So we're dealing with a 6-carbon sugar, and the aldo part tells us that it has an aldehyde functional group in its straight chain structure. So glucose can convert into this structure. This is the straight chain structure. And you could see the aldehyde functional group here. So that is the linear structure of glucose. Now let's move on to answer choice B, fructose. Fructose and glucose are similar in some ways. They're isomers of each other. Both glucose and fructose, they have the same chemical formula, C6H12O6. They both have 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms. But the structure of fructose is different than glucose. Fructose forms a 5-membered ring. It still has 6 carbon atoms though. So this is the structure of fructose. So this is carbon 1, 2, 3, 4, 5, and 6. Now fructose is a ketohexose. It's still a hexose because it has 6 carbon atoms, but its linear structure has a ketone functional group as opposed to an aldehyde functional group. So I'm going to draw that real quick. So that is the linear structure of fructose. And here is the ketone functional group. And that's how it differs from glucose. So fructose is a constitutional isomer of glucose. They have the same chemical formula, but as you can see, the atoms are connected differently. We have two different functional groups here. Now, this is called D-fructose, and the reason for that is because the OH group here is on the right. If we were to switch these two groups, then it would be L-fructose if the OH was on the left side. Now, let's move on to galactose. Both glucose and fructose are monosaccharides of carbohydrates. Galactose, would you say this is a monosaccharide? That is, it contains only one sugar unit, a disaccharide, two sugar units, or a polysaccharide, where it has many sugar units. Galactose is a monosaccharide like glucose. It's made up of one sugar unit. I'm going to draw the straight chain structure of galactose. And notice how it differs from glucose. This is once again D-galactose since the OH group here is on the right. But notice that this hydroxyl group is different from this one. So this is carbon 1, 2, 3, 4, 5, 6. So glucose and galactose, they differ at carbon 4. Everything else is the same. They have the same functional group. They're connected the same way, but they differ in the way the elements are arranged in space. So these two monosaccharides, glucose and galactose, they're known as stereoisomers. They're connected the same way, they have the same chemical formula, but their atoms are arranged in space differently. Now, galactose is specifically called a C4 epimer because it's only different on the fourth carbon. So it's a C4 epimer. So we can eliminate answer choice C because that's another monosaccharide. So do you think the answer is D or E? Sucrose or ribose? What would you say? Now, ribose is a 5-carbon sugar. It looks like this. Ribose is an aldopentose. Like glucose, it forms an aldehyde functional group in its straight chain form, but it's a pentose because it has 5 carbons as opposed to 6. So penta means 5, 6 means hexa. Anytime you see Os, you're dealing with a sugar. Now ribose is it's a 1 sugar unit, so it's a monosaccharide. The answer is sucrose. Sucrose is composed of 2 sugar units. It's composed of glucose and fructose. So because sucrose has 2 sugar units, it's not a monosaccharide, but it's a disaccharide. Mono means 1, di means 2. So sucrose is the answer. Number 3. Which of the following polysaccharides is the structural building material of insects? Would you say it's starch, glycogen, chitin, cellulose, or maltose? Well, let's talk about it. So we said that monosaccharides contain 1 sugar unit. A disaccharide contains 2 sugar units. A polysaccharide has many sugar units. And a number of sugar units could be in the thousands. So that's a polysaccharide. Starch is a type of polysaccharide. It consists of glucose monomers. Starch is a polysaccharide found in plants, and it's used to store energy. A good example of starch would be potatoes. Now, starch consists of two types of polysaccharides. You have amylose and amylopectin. Now, what you need to know about these two is that amylose tends to be straight. Amylopectin has branches. But they both consist of the glucose monomer. So A is not the answer we're looking for. It's a polysaccharide, but it's not the structural building material of insects. Glycogen is similar to starch, but glycogen is found in animals. It's used to store energy as well, and glycogen is highly branched. So this is not the answer either. Chitin is the answer. Chitin is the polysaccharide material. It's the polysaccharide that is the structural building material of insects. So C is the correct answer. D, cellulose, is a polysaccharide that is found in plants. But this particular polysaccharide has a structural function. It's found in the cell walls of plants. Maltose is not a polysaccharide. Maltose is a disaccharide. So maltose consists of two glucose units connected to each other. Sucrose, as we mentioned before, is glucose and fructose. Another one, lactose. This is a disaccharide consisting of glucose and galactose. So those are the three disaccharides you need to be familiar with, but chitin is the polysaccharide that is the structural building material of insects. So that's it for this question. Number 4. Which of the following are monomers of proteins? Would you say it's monosaccharides, nucleotides, fatty acids, amino acids, or nucleic acids? Well, we know it's not monosaccharides. Monosaccharides are the monomers or subunits of carbohydrates. It's not nucleotides. Nucleotides are the monomers of nucleic acids. And nucleic acids, those are macromolecules. Fatty acids can join to form a triglyceride. A triglyceride contains 3 fatty acids and a glycerol molecule. So this is not really a monomer of proteins. The answer is D, amino acids. When you have many amino acids joined together, they will form a polypeptide. And as the polypeptide chain folds, you have a protein. So proteins are made up of amino acids. So D is the correct answer for this problem. Number 5. Which of the following structures represents an amino acid? Well, let's go through each one. Let's start with answer choice A. So, what is represented by answer choice A? This is a fatty acid, which is a type of lipid. Fatty acids, they contain a carboxyl group and a nonpolar hydrophobic tail. That tail consists of hydrocarbons, and hydrocarbons are nonpolar. They're hydrophobic. They don't mix well with water. The carboxylic acid functional group, that's a polar functional group. That part mixes with water. That's the hydrophilic part of the fatty acid. So answer choice A is not what we're looking for. Answer choice C represents a triglyceride. Triglycerides also fall under the category of lipids. A triglyceride contains 3 fatty acids Here's the first one, second, and the third one, and it's attached to a glycerol molecule. Now, this particular fatty acid is a saturated fatty acid. The reason why it's a saturated fatty acid is because there are no double bonds. This is an unsaturated fatty acid because there is a double bond. Now, if you have a fatty acid with multiple double bonds, then you have a polyunsaturated fatty acid. So that's the difference between a saturated fatty acid and an unsaturated fatty acid. It's whether or not if they have double bonds. Answer choice D is a steroid molecule. Anytime you see 4 fused rings together, that tells you it's a steroid, which falls under the category of lipids. These molecules are also nonpolar. They don't mix well with water. Whenever you have a hydrocarbon, whenever you have a molecule that consists of carbon and hydrogen, it's going to be nonpolar. So all of this is the nonpolar part of the steroid molecule. The only part that's polar is the alcohol functional group. Whenever you see an OH bond or a CO bond, that's the polar region of the molecule. If you see a CH bond, that's nonpolar. So I'm just going to write that down. The CH bond is nonpolar. Let me put NP for nonpolar. But if you see like an OH bond, a CO bond, or like a CN bond, those bonds are polar. And also the NH bond, that's a polar bond as well. Now, for answer choice D, this particular steroid molecule is known as cholesterol. B is the answer we're looking for. B represents an amino acid. So here we have a chiral carbon, that is a carbon with 4 different groups. And a typical amino acid will have this carbon. It's going to have a carboxyl group or a carboxylic acid functional group. And it's also going to have an amino group, a hydrogen atom. But the part that differs in every amino acid is the R group, which is here. This is the part that changes. Everything else, every other amino acids will have these 4 things: the chiral carbon, the hydrogen, the carboxyl group, and the amino group. Now, when the R group is a methyl group or a CH3 group, we have the amino acid called alanine. Number 6, which of the following represents a nonpolar aromatic amino acid? So we need to analyze the R groups. So for answer choice A, notice that the R group only contains hydrocarbons. There's no OH bond or NH bond. So this is a nonpolar amino acid. Looking at answer choice C, analyzing the R group, we can see that there is a carboxylic acid functional group. So this is an acidic amino acid, and the carboxyl group is polar, so this is also a polar amino acid. So we can eliminate answer choice A and C. A is nonpolar, but it's not aromatic. Answer choice B, notice that we have an OH group. So that makes it a polar amino acid, which we're not looking for. Now, answer choice D is the answer we're looking for. The first thing is that it only contains hydrocarbons, which means it's going to be nonpolar. Second, we have a benzene ring. So that is an aromatic ring. So answer choice D is a nonpolar aromatic amino acid. This is called phenylaniline, abbreviated PHE. Answer choice A, this is known as valine, abbreviated VAL. B is called serine, the abbreviation is SER. And for C, that is aspartic acid, abbreviated ASP. Number 7. Which type of reaction occurs when two amino acids join together to form a peptide bond? Would you say it's a dehydration synthesis reaction, an oxidation reaction, a hydrolysis reaction, a reduction reaction, or none of the above? Well, let's talk about what each one means. An oxidation reaction occurs when there's a loss of electrons. A reduction reaction occurs when there's a gain of electrons. For this particular situation, none of those reactions will apply. Now let's talk about dehydration synthesis and hydrolysis. So when you think of the word synthesis, what do you think of? You're synthesizing something, you're building something. So let's say if we were to take two smaller units and combine it to form a bigger unit, let's say AB, this would be a synthesis reaction. Now, when you hear the word dehydration, what do you think of? If you drink a glass of water, you're going to be hydrated. You're adding water to your body. But let's say if you ran a marathon and you're sweating, you've lost a lot of water. You're dehydrated. So dehydration has to do with the loss of water. In this case, water is going to be on the right side. So this would be a dehydration synthesis reaction. You're building something by losing water. Hydrolysis is the reverse. Hydro means water, lysis means to split apart. So here we have the molecule AB, and we're going to add water to break apart AB. So that bond is going to disappear. We're going to break the bond using water, and we're going to split apart AB into A and B. So that would be a hydrolysis reaction. So a dehydration synthesis reaction will form a bond by means of the loss of water, whereas hydrolysis will break a bond by means of putting water into the reaction. Now, number 7, we're trying to form a peptide bond. We're not trying to break one. So that's going to be a dehydration synthesis reaction. Now I'm going to give you an example. So let's say this is the first amino acid. Here is the carboxylic acid group. Here is the R group. And we're going to join it to another amino acid. So I'm going to put R2 for a different R group. So this is a dehydration synthesis, I said that wrong, a dehydration synthesis reaction. So first we're going to lose water. We're going to take out H2O, 2 hydrogens and 1 oxygen atom, and then we're going to combine these two units together. And so here is the water molecule that was removed by creating the covalent bond. So we created a carbon-nitrogen bond here. So this is the amide linkage. You can see that's an amide functional group. And this represents the peptide bond, which is a type of covalent bond. So anytime 2 or more amino acids join together, they will always form a covalent peptide bond, and they will create an amide functional group. So that is a dehydration synthesis reaction. We took 2 smaller monomers and combined it into a bigger molecule, and we also lost water. So that's it for number 7. Number 8, which of the following is not a protein? Would you say it's hemoglobin, myoglobin, keratin, collagen, or testosterone? Hemoglobin is a protein that carries O2 in the blood. Myoglobin is a protein that carries oxygen or O2 in muscle tissues. Keratin is a protein that is found in hair and nails. It's a structural protein. And collagen is also a protein that is found in cartilage, tendons, ligaments, and skin. The answer is E, testosterone. Testosterone is not a protein. Rather, it's a hormone. It's a steroid. Here's the structure of testosterone. Like cholesterol, it has 4 fused rings, which makes it a steroid. It has a hydroxyl group, a ketone functional group, and it has a few methyl groups as well. Let me add the stereochemistry as well. So that's the structure of testosterone. It is the primary male sex hormone. So anytime you see 4 fused rings, know that this is a steroid. So that's it for number 8. Number 9. Which of the following is not an enzyme? Enzymes are proteins, and you could easily identify an enzyme by looking at the suffix of the word. If you see a word ending in ase, it tells you that you're dealing with an enzyme. Lactase is an enzyme that converts lactose into glucose and galactose. This enzyme, it breaks down lactose into those simple monosaccharides. Protease is an enzyme that breaks down proteins into amino acids. Amylase is an enzyme that catalyzes the breakdown of starch and glycogen. Think of starches like amylopectin and amylose. Amylase can break down amylose and amylopectin into its glucose units. Lipase, think of lipids. Lipase is an enzyme that breaks down fats like triglycerides into fatty acids and glycerol. Myosin is the only one that is not an enzyme. Whenever you see the ending in, it's a good indication that you're dealing with a protein. Think of the other proteins that we've considered, like hemoglobin or myoglobin or keratin. So when you see the suffix in, not always, but generally speaking, you're dealing with a protein. And ACE tells you that you're dealing with an enzyme. Number 10. Which of the following is not an amino acid? Is it polymerase, albumin, ferritin, insulin, or histidine? Well, we know that polymerase is not going to be the answer. We can see the suffix ase that tells us that we're dealing with an enzyme, which is a type of protein. Now, polymerase is an enzyme that synthesizes nucleic acids such as DNA and RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. DNA is double-stranded, whereas RNA exists as a single strand. Albumin is a protein that maintains the osmotic pressure of the blood. Ferritin is a protein that stores iron in the body, and insulin is a protein / peptide hormone that regulates the glucose levels in the blood. Notice that proteins, they end with the suffix in. Albumin, ferritin, insulin. In the last problem we said myoglobin, hemoglobin. Think of the proteins actin, myosin. These two proteins, they are involved in muscle contraction. but you can see they have the same suffix in. So when you see that, it's a good indication that you're dealing with a protein. Now, the majority of amino acids, they end with the suffix "ene." So think of valine, which we've considered earlier, serine, tyrosine, alanine. Most, not all, but most amino acids end with the suffix "ene." So that's a good way to determine if you're dealing with an amino acid or if you're dealing with a protein or an enzyme, which is also a protein. So sometimes the name gives you a clue as to what the answer is. So that's it for number 10. Number 11. Which of the following statements is false? So let's look at answer choice A first. Table sugar is composed of sucrose. Is that a true statement or is that a false statement? This is a true statement. B, glucose is the main source of energy for cells. This is also true. Glycogen, starch, they're all composed of glucose monomers. C, triglycerides are used for long-term energy storage. This is also true. For instance, let's say if a person decides to fast, if they decide not to eat for a while. The first source of energy that your body's going to look to are your carbohydrates, your sugars like glucose, your stored carbs like glycogen. That's the first thing that's going to go. Now, within a day, your body's going to run out of glycogen. Now, once you burn out your carbs, the next thing that your body's going to look for energy are your triglycerides, your fats. And if you drink plenty of water, a regular person can go days without needing food. Triglycerides, they can provide you with the energy you need for many days without having a meal. And so triglycerides, they're very useful for long-term energy storage, especially during periods of famine. So fats are not always a bad thing. Now, what about D? Saturated fatty acids are liquid at room temperature. Is that true or false? So this is the false statement. That's the answer we're looking for. A saturated fatty acid, which, as we mentioned before, does not have any double bonds, these tend to be solids at room temperatures, not liquids. A good example would be like butter. Now, there's two types of unsaturated fatty acids that you need to be familiar with. The first one are the trans fatty acids. You probably heard about that in the news, that these are not good if you have them in your diet. The other type, which includes the vegetable oils, these are usually better for your health. These are the cis fatty acids. They have a cis double bond. And because of this kink in the structure, the way it bends, these types of fatty acids tend to be oily. They tend to be liquid at room temperature. So think of like vegetable oil or olive oil. They have this unsaturated cis double bond. Now here's a way in which you could distinguish the cis from the trans double bond. If you were to draw the hydrogen atoms, notice that the hydrogen atoms are on the same side on the double bond. In the case of a trans double bond, the hydrogens are on opposite sides across the double bond. So that's how you could distinguish whether you have a trans fatty acid versus the regular cis fatty acid. Now, answer choice E, we know has to be a true statement. But let's talk about it. Cell membranes let me get rid of this first. Cell membranes are composed of phospholipids. We know that has to be true. So here is a simple way of drawing a phospholipid. A phospholipid has a polar head and two nonpolar tails. And they are used to form the cell membrane in a cell. This is known as the phospholipid bilayer. You have two layers composed of phospholipids. In the interior of this phospholipid bilayer, this is the hydrophobic region. That's the nonpolar region. That's where your oils will prefer to be. Water doesn't want to be dissolved in that area. This region is the polar region. That is also the hydrophilic region. And so water will be outside of that area and also below it as well. But water doesn't want to stay in the interior of that region. So that's the phospholipid bilayer, which forms the membranes of cells. Now a phospholipid looks like this. So it consists of a glycerol molecule, two fatty acids, one of them is saturated and the other one is unsaturated. Now it also contains a phosphate group. And these are three methyl groups attached to the nitrogen atom. Whenever nitrogen has 4 bonds, it's going to have a positive formal charge. So that's the structure of a typical phospholipid. So this phosphate group, along with the nitrogen in the 3 methyl groups, that is the polar head of the phospholipid. So that's the hydrophilic region. And that's the part that is water soluble. The other part of this molecule, this is the hydrophobic region. So this represents the two tails of the phospholipid. This is the part that's nonpolar. It's water insoluble. It doesn't mix with water. So just make sure you're aware of that. So to review, if you have a test, just remember a phospholipid is composed of the glycerol molecule, the phosphate group, and two fatty acid tails. One of them is saturated and the other is unsaturated. Number 12. Which of the following statements is false? Let's begin with answer choice A. DNA is the macromolecule that stores the genetic information of a cell. Is that true or is that false? This is a true statement. DNA is a nucleic acid. It stands for deoxyribonucleic acid. And yes, it definitely stores the genetic information of a cell. It tells the cell what type of proteins to make, when to make them, and other stuff like that. Now, answer choice B. Enzymes are proteins that speed up chemical reactions. That is true. An enzyme can catalyze a chemical reaction. It can make it go faster by lowering the activation energy. Another way to speed up chemical reactions is to raise the temperature, but the human body operates at an ideal temperature of 98.6 degrees Fahrenheit. So you don't want to speed up chemical reactions by increasing the temperature. Thus, enzymes are very useful. Part C. A nucleotide consists of a phosphate group, a ribose sugar, and a nitrogenous base. This is also a true statement. So this would be the 5-carbon ribose sugar. This would be the phosphate group. And then we would have the nitrogenous base. Now, this is a simple representation of a nucleotide. The exact molecular structure will vary. Now, there's 5 nitrogenous bases that you need to be familiar with. These are adenine, thymine, guanine, cytosine, and uracil. Now, what you need to know is that uracil is found in RNA, but not in DNA, whereas thymine is found in DNA and not RNA. Now, let's move on to part D. Lipids provide thermal insulation for animals in cold weather. This is a true statement. Lipids have many functions. They can be used for long-term energy storage. They can be used as hormones in the case of testosterone and estrogen. They're used to make the membranes of cells in the case of phospholipids. And they can also provide thermal insulation for animals. So animals with a lot of body fat, this will help them to stay warm in the wintertime. Now, E has to be the answer we're looking for. This is the false statement. Triglycerides are not soluble in water. And let's talk about that. So first, let's talk about fatty acids. Earlier in this video, I mentioned that fatty acids have a polar head and a nonpolar tail. Now, even though this part is water soluble, the bulk of the molecule consists of hydrocarbons. And so overall, because of the size of the nonpolar tail, this molecule by itself has a very, very low solubility in water. Only a very small amount of it will dissolve in water. So for the most part, you could say it doesn't mix well with water. So it's relatively insoluble. Now, even though we have an OH bond, typically when you see an OH bond, that's a special case of hydrogen bonding, and OH bonds are very polar. They can increase the solubility of a molecule in water. But because of this huge hydrophobic region, it's not enough to make the whole molecule soluble in water. So the solubility of fatty acids in water is very, very, very low. Now, in the case of a triglyceride, keep in mind the triglycerides have a glycerol molecule and 3 fatty acids. Let's add an unsaturated fatty acid, and let's add a polyunsaturated fatty acid. So notice that we have no OH bonds in this region. The CO bond is slightly polar, but it's not as polar as an OH bond. Because there's no hydrogen bonds, this molecule overall is nonpolar. You have 3 huge nonpolar tails and no OH bonds in this region. So triglycerides are not soluble in water. Number 13. Which of the following molecules is not a lipid? Go ahead and try this. So looking at answer choice A, this is known as a terpene. Specifically, this is called isoprene. This is a type of lipid. So we could eliminate answer choice A. B, we could see that we have the 4 fused rings. So this is a steroid, which is also a type of lipid. This is specifically called estradiol, one of the major components of estrogen, the primary female sex hormone. Now, answer choice D, this is known as a prostaglandin. It's also another type of lipid. Prostaglandins typically have a 5-carbon ring and a total of 20 carbons. So if we were to count it, this would be, let's call this 1, 2, 3, 4, 5, 6, 7, and then 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. As you can see, there's a total of 20 carbons. I'm not counting it in any special order. I just want you to see that there's 20 carbons in total, and it has a 5-carbon ring. So that's another type of lipid. Now, answer choice C represents an amino acid. Here we have our chiral carbon, which has a hydrogen. It has a carboxylic acid functional group, an amino group, and this right here is the R group. So let's redraw it in a way that is more familiar. So let's start with the chiral carbon. Now, we have the amino acid in its Zwitter ion form. If I take a hydrogen and put it here, it's going to look like this. So here's the carboxyl group of the amino acid. Here is the amino group. This is the hydrogen. And then the R group would be here. It has a total of 3 carbons and an amide functional group. So this particular amino acid is called glutamine. So anytime you see a carbon atom that is connected to a carboxylic acid functional group and an amine, you're dealing with an amino acid, the monomer of a protein. Number 14, which of the following nitrogenous bases is not a pyrimidine? Now you need to know that pyrimidines, they contain 1 ring. So answer choice A is a pyrimidine, C is a pyrimidine, D is a pyrimidine. All of these are nitrogenous bases. Now B has 2 rings. B is what is known as a purine. Answer choice A is the nitrogenous base called thymine. C is uracil. Keep in mind, uracil is found in RNA and thymine is found in DNA. D is cytosine. Notice the difference between thymine and uracil. Thymine has a methyl group, uracil does not. Besides that, everything else is the same. Now, cytosine differs from uracil in that instead of having this carbonyl group, you have an NH2 group. So that's the difference between cytosine and uracil. And also the presence of this double bond here. So cytosine has 1 oxygen atom, whereas thymine, uracil, those are pyrimidines with 2 oxygen atoms. So to review, the pyrimidines such as thymine, uracil, and cytosine, they have 1 ring. The purines such as guanine and adenine, they have 2 rings. Now this particular purine for answer choice B, which is the answer by the way, this is called guanine. So that's it for this problem. Number 15. Which of the following statements is/are true? Number 1. Macromolecules such as proteins may contain the element sulfur. Is that true or is that false? It turns out that this is a true statement. Proteins are made up of amino acids. Amino acids are the building blocks of proteins. They're the monomers of proteins. And there's one particular amino acid that contain the element sulfur. There's another one too, but I'm going to draw one of them. So one of the amino acids that contain sulfur is known as cysteine. The R group is CH2SH, so it has a thiol functional group. This amino acid is abbreviated CYS. So if a protein has this particular amino acid in it, which many proteins do since they tend to contain thousands of amino acids, then yes, they may contain the element sulfur. Number 2, the element phosphorus can be found in nucleic acids. Is that true or false? Now, a nucleic acid has 3 components. It has a ribose sugar, and it has a phosphate group, and it has a nitrogenous base, such as the purines or the pyrimidines. Now, because of the phosphate group in the nucleotide, which are monomers of nucleic acids, number 2 is a true statement. So all of the nucleotides contain phosphorus. So nucleic acids such as DNA and RNA, which are polymers of nucleotides, they will all contain the element phosphorus. So anything that don't have 1 and 2, we can eliminate. So we can eliminate answer choice D and E. Now let's focus on the ribose sugar. There's a hydroxyl group on carbon 3. This is carbon 1, 2, 3, 4, 5. The ribose sugar is a 5-carbon sugar. Now, in RNA, which is known as ribonucleic acid, it has an OH group on carbon 2. So that would be the case for RNA. Now for DNA, the situation is different. DNA stands for deoxyribonucleic acid. So we're lacking an oxygen in the ribose sugar in DNA. So in DNA on carbon 2, you have a hydrogen instead of an OH. So just keep that in mind. And that's how DNA differs from RNA. The OH group is replaced with an H. Now let's move on to number 3. Lipids do not contain the element phosphorus. Is that true or is that false? Number 3 is true for most cases. Most lipids do not contain the element phosphorus. For example, fatty acids only contain carbon, hydrogen, and oxygen. The same is true for triglycerides. They only contain C, H, and O. I'm not going to draw all of it, but just want to help you to see that. Even steroids with the 4 fused rings, they only contain carbon, oxygen, and hydrogen. Now, we do have phospholipids. If you recall, a phospholipid has the glycerol molecule, 2 fatty acids, one of which is unsaturated, and it contains a phosphate group. So phospholipids are lipids that do contain the element phosphorus. So based on that, number 3 is not a true statement. It's false. There are some lipids that contain the element phosphorus, particularly phospholipids. Number 4, nitrogen can be found in nucleic acids, monosaccharides, and proteins. This is true for nucleic acids. A nucleic acid will have a nitrogenous base. And as we've considered the purines and the pyrimidines, they contain nitrogen atoms. So you can find nitrogen in nucleic acids. You could also find nitrogen in proteins. Proteins consist of amino acids, and every amino acid will have an amino group, which contains nitrogen. However, monosaccharides, like glucose, fructose, they don't contain nitrogen. Glucose is C6H12O6. Galactose, fructose, they all have that chemical formula. So monosaccharides contain carbon, hydrogen, and oxygen, not nitrogen. So number 4 is not correct. That's a false statement. So the answer is A, 1 and 2. Those statements are true. Number 16. Which of the following statements is false? Let's start with A. Amylose is a polysaccharide consisting of glucose monomers connected by alpha-1,4 glycosidic linkages. Is that true or false? This is a true statement. Now, before we go into why that's the case, we need to talk about a few things. In its cyclic form, glucose can exist as alpha glucose, or it can exist as beta glucose. As you can see, the hydroxyl group is pointing up in its beta form, and it's pointing down in its alpha form. Because glucose can convert into a straight chain form, it can switch between its alpha and beta form. And when it does that, this process is known as mutarotation. The carbon atom, where it can switch between its alpha and beta forms, that carbon atom is known as the anomeric carbon atom. Now, amylose is basically a straight chain polymer of glucose monomers. And we said that amylopectin is branched. Let's talk about why that's the case. So in amylose, this is the type of linkage that we have. This is carbon 1, 2, 3, 4, 5, and carbon 6 is up here. So notice that the linkage is between carbon 1 and 4. And because the bond goes down, we have an alpha linkage. So we have an alpha 1,4 glycosidic linkage in amylose. And that's why amylose is straight. But now let's extend this a bit. In amylopectin, the situation is different. It forms branches. Let's see if I can fit this somewhere. So in amylopectin, notice that we have, this is carbon 6 by the way, an alpha 1,6 linkage. The alpha 1,6 linkage in amylopectin explains why there's branching. So the alpha 1,4 linkage creates monomers in a straight chain. You have that in amylose. Amylose only has the alpha 1,4 linkage. Amylopectin has both the alpha 1,4 linkage and the alpha 1,6 linkage. Just keep in mind, this particular glycosidic linkage is responsible for the branching that we see in amylopectin. So that's a brief review into the bonding found in amylose and amylopectin. Now let's talk about the bonding in cellulose. So for answer choice B, it says that cellulose is a polysaccharide consisting of glucose monomers connected by beta 1,4 linkages. And this is true. So instead of the bond going down in the case of amylose, it's going to go up in the case of cellulose. So this is still carbon 1 on the ring to the left. On the right, we have carbon 4. So it's still a 1,4 linkage, but we're dealing with a beta linkage. So it's going to be a beta 1,4 glycosidic linkage in cellulose. So those are some things that you want to commit to memory, or sometimes it's just better to understand it for the test. So you have a beta 1,4 linkage in cellulose, an alpha 1,4 linkage in amylose, and in amylopectin, it's alpha 1,4 and alpha 1,6. Now let's move on to part C. The primary structure of a protein is determined by its amino acid sequence. This is a true statement. That's one of those things you need to know. And for D, chaperonins are proteins that help new proteins to fold properly. That is also true. So we know that E has to be the false statement. But let's talk about it. The tertiary structure of a protein is characterized by formation of localized structures such as the alpha helix and the beta pleated sheet. This is true for a secondary structure and not the tertiary structure. So let's review this from the beginning. The primary structure of a protein is determined by its amino acid sequence. The secondary structure of a protein is based on the alpha helix structure that it forms and the beta pleated sheets. Now, the tertiary structure of a protein is based on how the polypeptide chain folds. So it has to do with the folding of the chain and its 3D structure. Now, sometimes you may have a protein with multiple subunits, multiple polypeptide chains coming together to form a functional protein. So thus, that would represent the quaternary structure. So if you have a protein with multiple polypeptide chains, then that combined structure would be the quaternary structure. In the case of hemoglobin, hemoglobin has 4 polypeptide chains. It has 2 alpha subunits and 2 beta subunits. So those 4 chains combined would form the quaternary structure of hemoglobin. So those are the 4 levels of structures that you need to know as it relates to proteins. So that's it for this video. For those of you who like it, don't forget to subscribe, and thanks again for watching.